Welcome to our lecture on Autacoids. So this is our first lecture for this semester, and in here, in this presentation, we are going to define Autacoids, and we are also going to enumerate the different Autacoids that are of veterinary significance, and we are also going to enumerate the different functions of these Autacoids. What are Autacoids? So the term Autacoid is derived from two Greek words, autos meaning self and akos meaning remedy or healing substance. Autacoids are locally acting hormone-like substances produced by a wide variety of cells in the body having intense biological activity, which act briefly at the site of synthesis and release. So they act, for example, on adjacent cells. Autacoids are also known as tissue hormones or local hormones. These are formed, released, and inactivated within tissues. So autacoids are usually vasoactive. So when we say vasoactive, now this is a substance that has an effect now of either increasing or decreasing the blood pressure. So it has an effect on the blood vessel. It can either increase or decrease the diameter of the blood vessel, thus in also increasing or decreasing the blood pressure as well as the heart rate. And of course, autacoids are a very important mediators of inflammation. They facilitate the development of inflammation. So basically, autacoids again are also known as hormones or tissue hormones, but they differ from the true hormones in the following ways. So number one, hormones are produced by specific cells in the body. For example, oxytocin is produced by the posterior pituitary gland, rather the hypothalamus. No, they are produced by the hypothalamus and they are released by the posterior pituitary. We also have the thyroxine. So thyroxine or T4 is produced by the thyroid gland. Now we also have insulin. Insulin is produced by the pancreas. So hormones, the true hormones, are produced by specific cells in the body, whereas the autacoids are produced by a multitude of cells in the body. They are also transported through circulation to act on distant target tissues. So, of course, another property of the hormones is that upon being released from its site of synthesis, it must first travel through the bloodstream and it will act on a target tissue that is distant from its site of synthesis. For example, in the case of oxytocin, for example, it is produced by the posterior pituitary and it must first travel towards the posterior pituitary. Then from the posterior pituitary, it must travel by the bloodstream towards for example, the mammary gland. So generally, the hormones will act on the distant target tissues, whereas in the case of the autacoids, they act on the area or the tissues that are near the site of synthesis. Another important characteristic of the autacoids is that again they are formed, released, and also they are inactivated within tissues. They have a brief duration of action, whereas for the hormones they have a longer duration of action. Autacoids can be classified according to different parameters, and one of that is via its chemical structure. So we have three general types of autacoids based on their chemical structure. The first are the amine autacoids. So they are known as amine autacoids because again they are derived from amino acids. Examples of this are histamine and 5-HT or serotonin. So again, the other name of serotonin is 5-hydroxytryptamine. Another type of autacoids are those that are derived from lipids. Examples of this are eicosanoids. So when we say eicosanoids, these refers to the prostaglandins, the leukotrienes, and the thromboxanes. We also have the PAF or the platelet activating factors. All of these are derived from the membrane phospholipids. We also have those autacoids that are derived from peptides, and these are the plasmakinins, now the bradykinin and the kallidin. We also have the angiotensin, the VIP or the vasoactive intestinal peptide, and the substance P. Another way of classifying the autacoids is based on their origin. So the first is the precursor molecules in the plasma. So they are derived from precursor molecules in the plasma, and under this we have the bradykinin, kallidin, and the angiotensin. We also have those that are preformed and stored in the cell. And this includes, for example, histamine, 5-HT, VIP, and substance P. So, for example, in the case of histamine and 5-HT, they are mainly derived from the white blood cells. For example, we have the mast cells, the basophils, no, as well as the eosinophils. We also have the precursor molecules and the cell membrane phospholipids. So they are basically derived from a molecule that is present in the cell membrane, particularly in the phospholipid component. And examples of this are the eicosanoids, the prostaglandins, the leukotrienes, thromboxanes, as well as the platelet activating factor, or the PAF.